Okay.
So I guess we're going to start. I'm going to say got it, and I know it's being recorded. Is this your computer? Is Mario's?
Sarah's got it upstairs. She's good.
Oh, we're good? Okay. So welcome to the CVRTI seminar. We have a distinguished external speaker today. This is also our last seminar for 2021. And so it's really an auspicious time. We, just by way of advertisement, we restart next month in January with RIPS, right? Starting off. And our RIPS talkers are Mohammed Khan from the Dazo Group and
Lindsay Roop from Cloud Lab.
oh, beautiful. Okay, great. The one warning I got from Tara is that if, from those of you who are on the web, sorry, I should talk into that, not the computer. I'm getting used to it. You send your questions to Derek and Derek will read them out.
Just in the chat box.
Just in the chat box, okay?
I'll read them.
And so I don't know, Mario, if you know the owl. I will introduce you later. Yeah, yeah.
We hit it right off.
So this owl is great because it lets us do hybrid meetings. So the majority of folks are actually online. And the owl has a presence. Sort of you'll see there it has a banner where you can see the room, sort of 360 degrees with a focus of view, and then everybody else basically a Zoom session, and then they can you'll share the screen, and that's shared. And so today is an auspicious reason for multiple reasons. One, just having Dr. Delmar here is reason enough. Two, we have started, and I want to thank Stavros Dracos a lot for helping kick this off, a joint seminar for our really distinguished speakers, a joint seminar with cardiology. And so Dr. Delmar is going to obviously give the talk today, now, and then tomorrow we'll give cardiology grand rounds. And so it takes a special individual to not just give two seminars where the slides aren't completely repeated, but it also takes a special individual to address two different types of audiences. And so that's reserved for only those who can pull it off. So thank you very much. We'll be actually the inaugural voyage into this sort of joint approach with cardiology and the CVRTI. I've known Mario, I don't know if you remember, but I think I may have first met you when we were talking about it when I was a student with Yoram. We were in New Orleans at an AHA meeting back in the day when we had meetings and there were 30,000 people at AHA. I'm not sure which is ever going to come back. We had gone out to dinner and you stopped by and said hello to Yoram. We were too small at the time. And after you left, Yoram said to me, Robin, watch Mario. I mean, by then you're already on faculty and on your way to stardom. And Yoram, as you know, doesn't do that lightly. And Yoram, of course, as usual, was prophetic because Mario is really one of the giants in connection 43 biology and in general cardiac electrophysiology, and has really taught us a lot of what we know from the channel biophysics to the cell biology to all the way to the bedside, which I think you'll probably cover even some of that today, I would guess, and tomorrow, excuse me, and tomorrow. So just by way of background, Mario did both his MD and his PhD, MD at the University Autónoma Metropolitana, right? And the PhD is a separate, the Sinvestav IPN in Mexico City, both, and then went to SUNY Upstate for the postdoctoral work and then stayed on there as an assistant professor, was a full professor at SUNY, right? And then went to Michigan for 4 years, was it? Was it really? Was you already sort of a chair position at Michigan? And then at 2010, if I remember correctly, to NYU. And so obviously as a full professor in 2010 and in 2015, the Patricia and Robert Martinsen Endowed Professor of Cardiology at New York University, and heads also the early translational program there with talk about sort of a company of the giants together with Dan Fishman, who is basically the other half of Connection 43 Biology of this country. And so I can tell you anything that we do in the lab, Mario has probably, or in reality has done first. You guys know, my lab knows this, you know, we do a lot of work with what we now call GJ120K. Well, guess whose group first described a small fragment happening endogenously in cells? The Delmar group, roughly 7 years before us. And so the same is true actually with organizational integrated disc programs. The same is true with cardiomyocyte biology in general. Now I think the focus is more and more on arrhythmogenic cardiomyopathy. Mario always gives the most elegant talks. I always learn not just from his literature, but from his talks. So I want to just a warm welcome for really a special time to have you here at the CVRTI.
Thank you. Thank you so much. Thank you. Thank you all for thank you all for having me. It is a real pleasure to be here. Robin has set the expectations a bit too high, so it is downhill from here. I actually have been in Salt Lake City before, and this is a number of years ago when Ken Spitzer was kind enough to invite me. So it is great to be back. A place that I have admired from the distance for a number of years, and that is great now to know that under the leadership of Robin and with the new faculty and with faculty that were here before, this is now the remaining or recovering its position as a leading center in cardiobiology. So thanks for being here. What I will show you today is work that has been done in my laboratory. I will, because of the setting, I will talk more about the more basic electrophysiology, and then tomorrow I will repeat some slides because the whole introduction is about the same, but then veer more towards what we think would be potentially translational. That being said, I will first acknowledge the people that have done the work while I spend my time visiting friends and having nice dinners. They are in the lab working really hard. And this is the team that has generated the data that I will present today in different papers and at different times. Most of what I will show has been done by these colleagues. I am also fortunate enough to have collaborations outside of NYU. I highlight the collaboration with the University of Copenhagen and Alicia Lundby, with whom we have been doing some cardiac proteomics that if I have time, I will show you. UW University in a translational study that is about to be actually accepted, is in press in the European Heart Journal. And that also with the folks in the ARVC program in Johns Hopkins. And then recently with John Nolan in Vanderbilt. So my laboratory is preoccupied with cases like this. This is the heart of a 16-year-old boy. He was playing soccer with his friends, seemingly healthy, active, enjoying life. All of a sudden, this kid stops, falls, it collapses. Efforts to resuscitate him are unsuccessful, and just minutes after behaving like a normal teenager, exercising, he's declared dead. At the MRI post-mortem shows that the heart has a normal structure in the septum, but the free wall of the right ventricle has been replaced instead of with a normal muscle mass with a fibroid and adipose tissue. The left is for the most part preserved, as you can see in the histology in a Trichrome staining. There is a lot of adipose, so the muscle is red. And instead of muscle here in the free wall, you see all of this white is adipose tissue, blue is collagen. And you can already see some collagen infiltrates on the left, but there is still a substantial amount of muscle, and that is why he is mechanically performing well. So this kid is diagnosed post-mortem with arrhythmogenic right ventricular cardiomyopathy. This is an inherited heart muscle disease that may result in arrhythmia, heart failure, and sudden cardiac death. Importantly, though, not all these components happen at the same time, and I will make that point further in the next slides. An estimated 50% of the cases with familial ARVC in which a gene is identified, they are linked to mutations in the gene coding for Plakophilin-2. Plakophilin-2 is a gene of the desmosome. As you may remember, desmosomes are intercellular adhesion structures that are holding the cells together. Best example to illustrate the function of desmosomes is the skin, which has a lot of desmosomes in the cells. If I stretch my skin, I don't rip the cells apart from each other. The cells stay together. They stay together because they have this Velcro that is holding them together. And importantly, when I release the skin goes back to its initial position. And that is because inside, the desmosomes are attached to intermediate filaments. In the case of the skin, keratin. In the case of the heart, desmin. The heart is a very flexible structure. It's a strong muscle, but it's a very flexible muscle. It has to stretch and contract and stretch over and over thousands, hopefully thousands of times. So PKP2 is then considered this very arrhythmogenic gene, even though it belongs to a structure that is not necessarily an electrophysiology structure. So from the beginning of the description of this disease, it hit our curiosity. Why would it be that a gene coding for a protein that makes Velcro between cells would actually be so heavily involved in an arrhythmia disease? Two things I want to indicate here. One of the diseases that the penetrance is low, and that has to do with complex genetics, and I will not go into details, but happy to discuss it later. The second is that Plakophilin-2 is indeed a gene of the desmosome, but what we have said, and we started this journey with, was by contesting the notion that Plakophilin-2 is only a gene of the desmosome. And you all know this, but the same can be said about pretty much every gene. The connexin gene, GJ genes, do make a protein that forms gap junctions. So connexins do form gap junctions, but that's not the only thing they do. They do other things. And these things can be as or more important as the ability of the connexin to form gap junctions. That is, genes are pleiotropic. They can accomplish more than one phenotype. And the point that we made back then, and we continue to make, is that Plakophilin-2 is a pleiotropic gene. What's a pleiotropic gene? It's a gene that instead of having only one phenotype associated with it, has multiple phenotypes associated with it. That's what pleiotropism means. Now, importantly, you can go between one phenotype and another and another and another one with time. And what we propose is that Plakophilin-2 is actually a pleiotropic gene. Indeed, Plakophilin-2 can be responsible for arrhythmogenic right ventricular cardiomyopathy. But this is not one word. In other words, the causes of the arrhythmia can be unrelated, although at the end all combines, to the causes of the right ventricular predominance and with the causes of the cardiomyopathy. All of the pieces together can be called ARVC, but you can have an arrhythmia without the other components, or a cardiomyopathy without the other components. So in fact, this is best described in this statement that Corrado, Mark Link, and Hugh Calkins did in this review in New England Medical Journal, and that is that ARVC typically becomes apparent between the second and the fourth decades of life. Clinically over disease is preceded by a preclinical phase
which is characterized by minimal or no structural abnormalities. That is the concealed disease. Now, importantly, sudden cardiac death happens during the concealed phase. The highest risk for these patients of dying and sudden unexpected cardiac death is under the concealed phase. Sudden cardiac death may be the first clinical manifestation of the disease. In fact, in this study in the Veneto region of Italy, 20% of deaths in young people and athletes were caused by a previously undiagnosed ARVC. So what we have tried to figure out is to identify that there is an electrical disease that is preceding the mechanical disease. And therefore, you cannot ascribe the arrhythmias that are life-threatening in these patients to a structural abnormality. Something is happening to the cells themselves. And I will go into detail on this by describing to you the phenotype of this mouse. Most of the data that I will present has been obtained in this model, which we generated now 5 or 6 years ago. It is a cardiac-specific conditional knockout of Plakophilin-2. So the Plakophilin-2 gene has been fluxed, and then the CRE is under the command of the myosin heavy chain, but only when we inject the mice with tamoxifen. So these are normal mice. They are born and they live without any problems. They do have their Plakophilin-2, but then at some point, which is going to be at 3-4 months of age of the mice, which is the young adult, we're going to inject them with tamoxifen. Tamoxifen is going to go and cause the knockout of the Plakophilin-2 gene at that moment in cardiac myocytes only. These mice are deficient in Plakophilin-2. And I know what you're thinking. Well, this is not a model of ARVC, because ARVC is a mutation and not a knockout. It is usually only in one allele and not in both. It doesn't happen only in cardiac myocytes, it happens everywhere, and it happens since birth, well, since before birth, since throughout the development and after. And you are right. Neither we nor anybody else, no matter what my colleagues say, can recapitulate the RVC. It has not been recapitulated to the extent that I just mentioned in mice, and certainly not in iPS cardiomyocytes, because iPS cardiomyocytes are embryonic at best, in a dish, without all of the environment that surrounds the heart. What we do is to characterize the endophenotype of a gene that when expressed, that expresses in the heart, and that when it is mutated, causes a disease. That's what we do. So I want to know what happens, what is the function of Plakophilin-2? And for answering that question, I am going to remove it from the genome at a particular point in time in a particular cell, and I am going to figure out what happens afterwards to then make hypotheses as to whether that is similar to what happens when you have a mutation in a patient. Okay? Well, so what happens? Well, this is a Kaplan-Meier curve, and during the first 4 weeks, nothing happens. So time 0 here is the time in which we inject tamoxifen. The mice are already young adults. And what you can see is that 2 and 3 weeks after tamoxifen injection, nothing happens. 4 weeks after tamoxifen injection, we are close to the point in which we start seeing death. These are spontaneous death, and the animals are dying because of end-terminal, end-stage failure. By about day 50, they all have died, whereas, of course, the controls remain normal. The controls are injected also with tamoxifen, of course. So I am going to talk mostly about 2 time points, because one of the nice things about having a mouse model is that you can control the initiation of the disease so that you can actually catch different snapshots as things start to become abnormal and then progressively get worse. So we went into 2 time points at which things are actually not causing death. What are these 2 points? At day 14 after reinjection, we do not see any structural disease in these hearts. This is a Trichrome staining, and what I have done here is exaggerate the contrast on the blue so that you can see if there is any collagen. Of course, there is collagen in the great vessels.
but in the hearts are structurally normal. If I wait one more week, then what I see is that there is quite a bit of fibrosis in the free wall of the right ventricle, approaching an apical aneurysm there. But the septum and the free wall of the left ventricle are pretty much in normal conditions. So this looks a lot like the heart of the patient that I showed you at the beginning. You have cardiomyopathy of right ventricular predominance. Now, is it arrhythmogenic? Yes, these mice are arrhythmogenic at this stage. These are 3 different time points, and I will address them later. But at 3 weeks after the injection of tamoxifen, out of 10 animals that we tested for an injection of isoproterenol and see how many PVCs they give us, well, we get 6 of them that gave us more than 100 PVCs in a 20-minute interval, and that is this bar. Whereas the control group, only 1 mouse gave us some extra systems. At 14 days, we really weren't seeing much, but just wait a day or 2, and you would think that this bar is clean, except this bar is reflecting the number of PVCs. But there are 3 mice of this group that didn't give us much PVCs. They went straight into polymorphic ventricular tachycardia, ventricular fibrillation, and death. So the mice do have a phenotype of an arrhythmogenic right ventricular cardiomyopathy. So I am going to describe first this point, the 14 days post-tamoxifen injection. The phenotype, and I will go more into details tomorrow actually, but the fact that you have a structurally normal heart and with isoproterenol or with exercise, you can actually induce severe arrhythmias, is reminiscent of another inherited arrhythmia disease called CVVT, catecholaminergic polymorphic ventricular tachycardia. These are caused mostly by mutations in the ryanodine receptor. And I show you this slide also because if I have time, I will go back to this slide for presenting some of our data. But in CPVT, what you have are, for the most part, you have mutations in the RYR2 channel. Just as a quick reminder of this, which of course you all know, if this is the T-tubule, this is the membrane of the cell, this is the junctional sarcoplasmic reticulum that forms a dyad with the T-tubule so that the calcium channel is right in front of the ryanodine receptor. During the normal cardiac cycle, upon depolarization caused by the sodium channel, you get calcium entry. Calcium is going to bind to the ryanodine receptor to cause what is called calcium-induced calcium release. The ryanodine receptor channels will open and there is calcium stored in the cisterna that will leave the cell to reach the sarcomere and elicit the contraction. That calcium will then be pulled back into the cisterna by a combination of SERCA2A under the control of a phospholamban protein. So we will refill the tank here. Some of the calcium will also be expelled if necessary by the sodium calcium exchange. So CPVT is caused by mutations in here. We decided to look at whether there would be calcium dysregulation caused by the Plakophilin-2 knockout in these mice. And first I show you that indeed these hearts, when you take them into a Langendorf and you do optical mapping, you do see arrhythmias that result in this case from rapid pacing. I will not go into the details of the colors, but it just makes the slide prettier, so I leave the colors there. But the important thing is that we do observe a significant amount of arrhythmias that seem to initiate in the right ventricle. When we studied the regulation of calcium in these mice, what we observed is that there is an increase in the spontaneous release of calcium from myocytes of the right ventricle. So structurally, the right ventricle looks normal, but molecularly, it does not. What we have observed is that if you look at the incidence of early after transients or delayed after transients,
by using conventional calcium imaging techniques. Then you look at the percent of cells with early or delayed after transients. Control cells from the left ventricle or from the right have a certain amount of PATs. Myocytes from the knockout on the left ventricle have about the same, but the occurrence of early and delayed after transients is much higher in the myocytes that come from the right ventricle of the PKP2 knockout mice. So the cells from the right ventricle of the knockout mouse have a high tendency to deliver early and delayed after transients, as well as spontaneous calcium waves. And in the following slides, I will use the same color code. So red is going to be data that comes from the knockout. Black is going to be data that comes from the control. And for the next slides, I am going to separate myocytes from the left and from the right. So how about the frequency of calcium sparks, this spontaneous calcium release that is occurring out of the ryanodine receptor channels? Well, similarly, if you look at the occurrence of sparks, for those of you that do not know the sparks or the calcium imaging, just follow me here. When you see green spots, that is because there are ryanodine receptor channels that are opening and spewing calcium into the environment that is detected by a fluorescent indicator. So you can just look at how many times do you see these events, these spontaneous calcium release events. And if you look at what the wild type myocytes do, you count a certain number per unit of distance and time. If you look at the myocytes from the conditional knockout from the left, they are about the same, occur with the same frequency. If you go to the right side of the control, you also have about the same frequency of sparks. But when you go to the myocytes of the right ventricle of the knockout, we see this very large increase in the frequency of calcium sparks. Similarly, if we then add isoproterenol to the soup, then all of these parameters increase, including the occurrence of the sparks in the right ventricle. So there is a higher tendency for right ventricular myocytes to release calcium from the ryanodine receptor. One of the reasons why you can release excessive calcium from the sarcoplasmic reticulum is because your cisterna are full. So if there's a lot of calcium inside the cisterna, when the RIR2 flickers eventually, you're going to release more calcium because you have a higher gradient. So we looked at the amount of calcium in the sarcoplasmic reticulum, what we call the SR load. And that is done by presenting to the cells caffeine, which will completely fully open the ryanodine receptor. And so the calcium will be emptied into the cell. We will detect that calcium transient as a big, large calcium spike, and we're able to measure the amplitude of that, which is a reflection of how much calcium was inside the cisterna. And what you can see here is, again, knockout or control have about the same amount of calcium inside the cisterna. But the right ventricular myocytes from the knockout have a larger amount of calcium stored inside. So there is an increase in the amount of calcium that is stored in one of the major compartments that store calcium in a cardiac myocyte. There are four major compartments that store calcium in a cardiac myocyte: the SR, the cytoplasm itself, the mitochondria, and the nucleus. I'm not going to show you data on the nucleus, but it follows the rest. And that is that when we looked at the other compartments, we observed that the cytoplasmic and the mitochondrial calcium content in the RV cardiomyocytes is increased. So you can see here, the cytoplasmic is increased and the mitochondrial calcium is increased. So RV myocytes have more calcium in every compartment. So when we were doing these experiments and getting this data,
I was in close contact with my friend Dr. Valdivia, who is an expert in ryanodine receptor and calcium homeostasis. And I say, where is that calcium coming from? And he said, well, it's got to be coming from outside because all of the compartments are full. So there's calcium coming from outside. So what would be the source of that calcium? And John Chul Kim, who was a postdoc that was doing these experiments, very bright guy, back now in his native South Korea, said, how about connecting hemichannels? I said, okay, please. I've been arguing with my colleagues that there are no connecting hemichannels in the living ventricular myocytes. It's not possible, you know. He said, let me try it. So of course, you know, never, moral story for all of you faculty and young people, never stop a postdoc from letting him try, either because they're going to hit the wall or because they're going to get something really cool. And in this particular case, he did hit something really cool. So he had to figure out a way of detecting the presence of connecting hemichannels in these ventricular myocytes. So what he did was to load the heart in Langendorf with lucifer yellow, which is a fluorescent marker that will go through hemichannels, and with a marker that will not go through hemichannels, will only stay in the intercellular space, and that's the red marker. He would then perfuse this, then fix the heart, remove the epicardial layer, and then take the either left or right free ventricular wall face down onto the stage of the microscope and look at the fluorescence. And what you can see is that there are some myocytes from the control that take some fluorescence. Some also from the left ventricle of the knockout. But he found a lot of myocytes that would have more fluorescence, so that they would take more lucifer yellow from the right ventricle of the knockout. He was happy with that. I said, you have to really convince us. And the only way in which I'm going to convince me is if you get the opposite result, if you cross the PKP2 knockout mouse with the Connexin 43 knockout mouse. That is not a trivial experiment. The reason is that these are also Connexin 43 knockout under the same pre-control. When you knock out Connexin 43 in adult heart, the animals live about 14 days, and then they die with severe arrhythmias. So we had to inject several animals that would last until day 14, which is when the PKP2 knockout effect was observed, so that we could see the knockout of both. And so 2 or 3 of 10 or 15 animals that we injected with this double crossing did get the Connexin 43 knockout. So in this double knockout, then there is no entry of lucifer yellow. He then he said, okay, let's do it with the heterozygote connexin so that the probability of getting some data is higher. And even with the heterozygote Connexin 43 knockout and full homozygote PKP2 knockout, we observed that the diastolic calcium went back down and the spark frequency went back down, although not all the way to baseline. To further make this point, he then tried a molecule that is being reported to close Connexin 43 hemichannels, and that is called GAP19. And you can see that the calcium transit magnitude that was higher in the RV myocytes is now normalized in the presence of GAP19. SR load is normalized in the presence of GAP19. Spark frequency goes down, although not completely, and diastolic calcium goes down. And the reason why spark frequency does not go down completely is because, I won't have time to discuss this, but there is a change in the phosphorylation state of RYR2 that is present at this stage. All right, so what do you think is going on? Well, this is a schematic that, like all schematics, are not anatomically correct, but makes the point.
This is from a review on European Journal of Heart Failure. And what is showing is that at the intercalated disc, you have mechanical junctions in the vicinity of the gap junction. Important thing here in this schematic is that for a gap junction plaque to be stable, we have known this for many years, for a gap junction plaque to be stable, mechanical junctions, effective mechanical junctions are necessary. If the cells are not mechanically stable, the gap junction plaque will not form. And so what we hypothesized here is that as the mechanical junctions become unstable, then the gap junction plaque also becomes unstable, but there are channels incorporated already in the membrane, hemichannels, and as the cells start to separate, these hemichannels will start to flicker, and the flickering of these hemichannels will allow the entry of calcium into the cell. We have done experiments, then we have implemented really my knowledge for the first time, but may not be the case, thick sectioning immuno-EM for connexin 43. And this is, you take a thick section of heart, and then you put that on a scanning electron microscope with a machine that cuts slices. Microtome, thank you. And so you take a scanning EM, cut the slice, you take this next scanning EM, cut the slice. But before, we have incorporated nanogolds coated with antibodies for connexin 43. So what you see here are single planes of three-dimensional reconstructions of gap junctions from heart tissue. And it's actually very nice that you can actually see that there is two parallel strings that are forming the gap junction plaque. You lose a little bit of the spacing in there, but you can see there and there two nice parallel strings, two connecting on one side and on the other, forming a parallel string. In the knockout, on the other hand, we observe these bulgings that are happening in the middle of the gap junction plaque, as though the plaque is separating. We think these are hemichannels that were together or were poised to be together with another connexin on the other side, but didn't make it. And these may be the ones that are active. In different sections, we have found actually, again, in the control, you see different planes, nice two strings that are running parallel to each other. But then in the knockout, we observe sometimes, we observe sometimes that there is only one of the sides. And these are different planes. So this is not that we did not catch the other side. As we go through all of the planes, we observe only one string instead of two. So we think this is the anatomical correlate of the functional data that I have shown you. I'm going to go now to the 21 day. So 1 week later, when we do have an arrhythmogenic cardiomyopathy of right ventricular predominance, and there we also observe, in this case, we're not separating right and left because the right ventricle is pretty damaged, so we cannot dissociate myocytes from the RV anymore. We are dissociating them from the heart, and you observe a very similar thing. You have an increased frequency of sparks, increased amplitude of the sparks, increased diastolic calcium. Very quickly, we have what is called increased EC coupling, and that means that we have a reduced calcium current, but the reduced calcium current is enough to induce a larger calcium transient. So even though there is less calcium coming in, the response from the SR is of a transient that is larger. There is an amplification in the system. We did RNA-seq in these mice, and we actually, one of the advantages of this model is that it's very reproducible. So when you look at the heat maps, each column being from one different mouse, you observe that there is very high consistency within the groups. And we got, fortunately, that the most prominent gene network is ARVC. But we also observe genes of the calcium signaling pathway being downregulated as expected. So one of the things that is clinically very relevant in ARVC is the fact that exercise increases the progression of the cardiomyopathy, the likelihood of arrhythmias, and the likelihood of sudden death. And it is not clear why are these things happening. Why is that increase in the probability of the disease or in the progression of the disease by exercise? All we know is that it is advisable for patients with a diagnosis of ARVC to reduce to the bottom the level of exercise. We wanted to then use this model to see whether we would see some relation between exercise and the ARVC. So we put the mice to train in the treadmill. So mice now, these mice, both control and the knockout, are run for 3 weeks. Then they are injected with tamoxifen, both the control and the knockout. Of course, the control does not cause a cardiomyopathy. And then they are run for 3 more weeks. So after a 6-week training period, we observe in the control mice changes that are expected from a trained animal. There is a minor but significant increase in left ventricular ejection fraction. And in the fractional shortening is on the edge, there is an increase in the left ventricular anterior wall thickness during systole and the posterior wall as well. And these are data that are coming out in the European Heart Journal in the next couple of weeks. So we decided to also study this particular animal. This is not PKP2 knockout. This is just a mouse that has exercised. And we decided to look at what is the RNA-seq data on these mouse. As you know, there is such a thing as the athlete's heart. So athletes that overtrain can actually develop a cardiomyopathy that has some reminiscent parameters to ARVC. So when we did the differential transcriptome of the exercised animals, so this is a differential mice that are run versus mice that are kept sedentary, we have a nice separation of upregulated and downregulated genes. And on the upregulated, you have an upregulation of genes that have to do with cardiac muscle contraction and genes that have to do with metabolic pathways, as you would expect. What we were very surprised to see is that in the downregulated genes, we actually found as the most prominent pathway, the ARVC pathway. So these are control exercised animals, and yet the downregulated genes come from the ARVC pathway. And I actually will flip back to here, and you can see that their position in the volcano plot: desmocolin, desmoglian, Plakophilin-2, and anchorin B, which is part of the intercalated disc complex. So what we decided to do is to compare the transcriptome of a mice that exercises with the transcriptome of a mice that loses PKP2 but is sedentary. So in this plot, this is in the x-axis, trained minus sedentary control mice. So this is going to be the log2FC of each one of the genes. Each point is one gene in the exercise control animals. And on the y is the comparison of the sedentary PKP2 knockout minus the sedentary control. So this gene is upregulated in the exercised animals control, and it is also upregulated by the loss of PKP2. This gene is downregulated by exercise, and it is also downregulated by the knockout of PKP2. And when you look at this complete map, you can see that in this Cartesian map, most of the genes are either on the first or on the third quadrant. In other words, there is a very high correlation between what happens to a gene when you exercise and what happens to a gene when you have a deficit in Plakophilin-2. If you then combine both deficits, then what you have is a drop in ejection fraction that happens to a much higher extent in the PKP2 mice. So if I take now the sedentary mice, this is the ejection fraction at 0, 14, and 21 days. If I exercise these animals, though, the ejection fraction drops much more because of the combination of the exercise and the loss of Plakophilin-2. So what we think is happening is that there is, and I borrow actually a term that may have been born here, and that is the repolarization reserve. Well, I am going to talk about a desmosomal reserve. When you exercise, the transcriptome of the athlete actually shows, mouse, I don't know if this happens in humans, the transcriptome of the exercise of the athlete mouse shows a drop in the expression of genes that code for desmosomal molecules, and in general for molecules of the intercalated disc. So if you are a normal mouse, not you, but if you take a normal mouse and you look at the abundance of desmosomal genes, say we are at this level. When they exercise, those levels may decrease, but still you are remaining above the threshold for the disease. If, on the other hand, you start with an animal that is already closer to the threshold for disease, when you exercise them, they drop past that threshold, and now they manifest a more acute disease. And we think this may be happening with humans. And indeed, those athletes that then develop an athlete heart may be athletes that by nature, in their own genome, they may actually have a low desmosomal reserve. They may already be on the verge of dropping too much close to the threshold for disease. And so even though they don't have one mutation or one particular gene, they may cross the threshold and then they go into apoptosis, cell death, and the phenotype of ARVC. Finally, I will tell you something that we haven't published yet, but also in the calcium regulation, exercise is making things worse. So this is animals that were trained and animals that are sedentary or controlled. You can see that the spark frequency also increases in these animals with training. The amplitude of the sparks also increases with training. The SR load is also higher in animals that have been training. So the whole phenotype just amplifies itself by exercise. We then decided to look with Alicia Lundby at the changes in the phosphoproteome of these hearts to see if there is something that is happening with exercise. And what we found, this is the overall result. This is the phosphoproteome volcano plot for the exercise animals that are controlled. This is the phosphoproteome in the knockout animals that exercise. And what caught our attention is the upregulation of the hyperphosphorylation of phospholamban and the hypophosphorylation of genes that have to do with the chelation of calcium in the sarcomere. So we think that what is happening with exercise is that there is a hyperactive phospholamban that is increasing the reuptake of calcium into the SR. At the same time, there is a hypophosphorylation of the genes that act to buffer the calcium in the sarcomere. And therefore, there is more binding of calcium in the sarcomere. There is a slower release of calcium from the sarcomere. And that increase in diastolic calcium is increasing the probability of EADs and DADs. So with this, I leave it to you as our latest
data, I have tried to take you through an experience of about 5 years of studying this particular problem of the relation between plakophilin-2 and calcium dysregulation and the role that this phenotype may play in the manifestations that occur with exercise. And with that, I will thank you very much for your attention and happy to address any questions.
We have a few, let me turn on the lights. As you give your talk, Mario, I'm just reminded of your ability to synthesize concepts, I think bar none. And actually, I haven't stayed current. I love where this is going. I have a few questions, but I thought I saw Joe's hand up and then we'll take turns. Yeah.
Yeah, thank you for the talk. I'm curious how you train the mice on the treadmill. So are they getting shocked or anything, or do you?
We actually, it's not trivial
Yeah.
because we do have an electric plaque at the bottom, but we actually have a postdoc, a very dedicated postdoc that keeps pushing that.
Right.
And now you start these experiments that are 6 weeks of running are very frustrating.
Yeah.
Because we start with
Yeah.
18 mice. Okay, so we take 18 mice. We have a treadmill of 6 rounds. So we do 3 rounds a day with a group of 18. And then we just keep following them. And we see that some don't want to exercise. They don't want to stay in the treadmill. They go back to the cage. And so from 18 mice, you finish with about 5 after the 6 weeks.
And then have you looked at things like cortisol? Because a lot of times the mice that get shocked in our experience, the cortisol levels are increased in the mice that exercise. So I was wondering if you think that there are other pathways specifically from getting shocked or getting hit to remain on the
So we
treadmill.
try them to not get shocked.
Okay.
We want them to get back into the treadmill by just using a spoon and push them back into the treadmill. And as I said, in some, this is gruesome, but some actually try to escape by going forward and their heads get caught in the band. So it takes a lot of patience. So these ends are not very high. When we were in the reviews of this paper, the statistics reviewer was asking us if we did our power calculation. So this is my power calculation. I have a very powerful postdoc, and he's gonna try this until we get enough data. That's my power calculation, because you really cannot predict. And the knockout actually is worse.
Right.
Because they are losing cardiac function. So they have to keep them. We do adjust the speed a little bit in the knockouts towards the final stages so that they can complete the time, even if the speed is not the same.
And roughly how much are you running? Like how far? Or how?
They run an hour.
Oh, okay.
They run for an hour, 5 days a week.
3 times a day.
Times a day.
So 1 time a day, go back to the cage.
Yeah.
Also because we run them in the day. I think that if we could run them at night, they would run better.
Sorry, one other question. I know your work really ties together everything about the calcium loads, increased diastolic calcium, but can you relate that to the increased fibrosis that you see later? So you have a great explanation for the reason why the arrhythmias happen before the fibrosis. And the fibrosis, you're not arguing is the cause of the arrhythmias, but like, why is the fibrosis?
Yeah, so let me clarify that. The clinical experience here from my colleagues is that the worst arrhythmias actually happens when you have more muscle mass. These patients then settle into a lot of monomorphic tachycardia. You can capture that in the Holters. They have a very high incidence of PDCs, and you can ablate them, actually. There are more and more centers that are ablating them. So that is where the structural component comes in. Now we know from work that dates back years that the more muscle mass, the higher the likelihood of fibrillation. So The most dangerous arrhythmias are happening not when the structural disease has settled, but before, because you have more myocytes that can generate re-entries and so on. Now, the cause of the fibrosis and of the fibroadiposes are very much, there are a number of studies showing that that they are consequence of aberrant intracellular signaling that activates pro-fibrotic genes in the myocytes, and like also in a distal way, activation of proliferative fibrosis in the fibroblasts that are resident in the heart. But I have not talked about fibrosis. It is obviously, the adiposis in the mice is virtually impossible to study because they don't develop adiposis, mainly because of the time. If we had more time, probably they would show the adiposis. At the transcriptomic level, they do show some activation of adipogenic genes.
Yeah, and then we're taking turns here. Did I see your hand up, Defiant, as well? Oh, okay.
Yeah,
Okay, good. And then, all right.
Yeah, conceptually very sophisticated. Yeah, thank you so much. I was just, going back to something Joe touched on, the calcium channels. So the decay is kind of slow, which probably suggests that reuptake is impaired and things like that. Yet, still calcium spot increased frequency. And calcium efflux is also reduced. So I'm just kind of wondering how would that work? So, the transient with the caffeine-induced calcium salt content.
So the SR has more, the calcium transient of the caffeine experiment increases.
Increases, yeah.
The amplitude of the calcium current
Is reduced.
is reduced.
Right.
The abundance of the transcript for CACNA1C is decreased.
Is decreased.
So
yes.
there is an increased amplification
I see.
in the diet.
Right.
And that is consistent with the changes in the phosphorylated state of RYR2 that we see.
Despite the fact we have reduced reuptake.
Exactly.
Right.
There is an increased eagerness of RYR2 for opening,
I see.
probably because of its phosphorylated state.
Right. And one more question. I'm sorry. So the PVCs you only see with response to ISO. So you don't see them spontaneously, which suggests that is that a limitation of the model? or
It's a
is that-
limitation of my lab because we don't have
Right.
a holder system.
I see. Yeah.
So we should just bite the bullet and buy a. I have been waiting for Glenn
Right.
to buy one, but might as well just buy one.
Sure.
Because indeed we don't have a measure of how much is spontaneously happening, especially when they are running,
Right.
which I suspect some of them stop running because they are having a lot of arrhythmias.
Well, so could sodium channel be altered here? or-
Yes.
Okay.
So,
Right.
yes, so we have previously shown, it's a very weird story with the sodium channel,
Right.
but in general, but not in this model specifically, there is a reduction in the amplitude of sodium current.
I thought that was a plant. I know very well what you're referring to. Wow, that's pretty good. Defiant.
Thank you very much. That's a wonderful talk.
Thank you.
I was thinking about this mechanism that you're describing, really intriguing in terms of the cardiomyopathy. And that seems to me that it would have two predictions as far as humans are concerned. One would be that if you look at cardiomyopathy in athletes versus non-athletes, the RBCs, that you can see differences in degree of cardiomyopathy as opposed to the burden of right ventricular failure or right ventricular fibrobody deposition. And I was wondering if that's true. And then the second thing is if the athletes of the RBC are more prone to these cardiomyopathy, would you see an actual decline in their fitness for whatever sport they're involved in prior to their presentation with some cardiac death? Would that be kind of the signal event in terms of actually predicting someone potentially at higher risk for mortality?
So those are very good questions. I don't have good answers. I will tell you what part of this study that is coming out. Actually, our first hypothesis when we started these experiments, the hypothesis that we wrote to the grant, is that we thought that there is an inability of the PKP2 deficient heart to activate compensatory transcript programs that compensate for excess demand. So
a regular athlete will have an increase in the dimensions of the walls. And we thought that that hypertrophic remodel transcriptional is not active in these models. That hypothesis didn't completely pan out, but it did to the following. We collaborated with Hopkins and the UTF groups that have the largest series of ARVC cases. And we asked them to compare, trying to match as much as possible two populations, athletes that don't have ARVC and athletes that come to be evaluated for the first time for a possible diagnosis of ARVC because they had syncope, that they were then examined for MRI and then found to have or diagnosed with ARVC. What are the dimensions of the walls? Equally trained. Both of them are equally trained. Similar age, same gender, same sex. And what we found is that the thickness of the wall of the patients with ARVC are not the same as the control athletes. The control athletes have a thicker wall than the ARVC patients. So something is already happening that is preventing these cells, and this is in patients, this is in humans. Something is happening that is preventing these cells from fulfilling the hypertrophy program. Can that be predictive of how bad is it going to be for someone to exercise? I would imagine, although they have not implemented that, but I would imagine that the way I am imagining this is that if you are exercising and you are putting an extra demand on the wall, an extra load because of the exercise on the wall, particularly the right ventricle, you need to maintain a good desmosomes to hold the cells together. If the cells do not hypertrophy and are not able to maintain the sufficient work for the increased load, the cells are going to start getting damaged and they're going to start breaking down. And that may be the beginning or one of the beginnings of the loss of the muscle mass, particularly in the
We're running short on time. I'm really sorry, but can we most folks are listening via Zoom. So I want to, out of respect to the Zoom participants.
One question then
Yeah,
from the chat. Sutat from Stavros Dracos' lab asked, do you have insight underlying the right dominance caused by PKP2 as opposed to left dominance? He says, my understanding is that PKP2 expression does not show right dominance. Is the left dominance Arrhythmogenic Cardiomyopathy? Does it have the same calcium uptake anomaly in the left ventricle? So why is this right-sided?
Right. So I think that the right-sided, as I was explaining, I think the right-sidedness is that the disruption starts with the loss of integrity of the desmosome, and because of the balance of forces, and here I am hand waving, but because of the balance of forces and stress of the free right ventricular wall is less tolerant to the weakening of the desmosome than the left. Do I have the data? Do I have the knowledge to actually test that hypothesis? No, because that requires a lot of someone that is really good at cardiac mechanics, and I'm not that guy. But
It's so much thinner, right?
yeah, exactly. The wall is much thinner.
Yeah.
And what I do know, a colleague of ours in Australia, Andre Lagache, showed me these images of ultramarathoners that they get MRI'd right after the finish line. So after running whatever 50 some miles, or then climbing a mountain and fighting a lion, and then they take their MRI. The right ventricle is this big. The amount of dilation of the right ventricle in a highly trained athlete is enormous. So my naive concept is that this in an athlete is actually putting an excess stress on the free wall, and this disruption of the desmosome is therefore happening first.
Thank you. Fantastic. I just, before, you're not running away, but I believe I hope you already had a look. This is sort of our CVRTI swag bag. Thank you very much, Diane. So in it is a history of the CVRTI,
Oh, great.
authored by none other than Ken Spitzer,
Wow.
as well as 2 hats, one sort of for the winter, for walking, if not the water front, then
Wow.
the
Thank you
or
so much.
the park. And then another is a cap. So thank you. It's really been an honor and a pleasure. And this is just the start of your visit. That's perfect. Thank you. Very much. All right, I'm sorry we ran out of time. In fact, I ran out of time for mine, so I'm gonna have to follow up